So good morning, everyone, and welcome to another Earth Institute Coffee Morning, the second of the new academic year. And again, we have a new member joining us this morning. It was really great to welcome you to the Institute, Darrin. And I know Darrin's only recently joined UCD. In January this year, he joined the School of Biology and Environmental Sciences from the University of Constance in Germany. But he hails originally from Texas, but hasn't been living there for many years. So he's gravitating to the European centre of gravity. But we're going to be hearing him talking about his work, which is a real interest to me. Particularly, I've had a long interest in traits and how traits work and whether they can be useful in identifying aspects of the way ecosystems work. And so Darrin's talk is about fishing for genome-to-ecosystem linkages in North America's aquatic endemicity hotspot. So thanks very much, Darrin. I'll hand over to you.
Okay, thanks so much, Taz. Thanks everyone for coming. What I hope to talk about is something that can be interesting to all of us. I'll be speaking about this endemicity hotspot in North America that's an aquatic oasis out in the middle of the Chihuahuan Desert of Mexico called Quatrocynicus. And what I normally do these days is actually study developmental genetics of things like teeth. And so what I hope to do is kind of mesh those things and provide some stimulating insight potentially in how you might mesh these things or not. So to get started here, first I'll acknowledge some of the people who've been very important in this. Francisco Garcia de Leon, who's been my longtime collaborator in Mexico, and several other people like Joel Chavez Campos and Axel Meyer Lab Group, who I was recently a part of before I moved here to UCD. And then a lot of my funding sources that provided me with the ability to do some of this work. So the reason I got interested in this system is because of this unique fish here. So this fish in the bottom left-hand corner is named Herikthes Menglia. And like most species of fish, it actually has two sets of jaws. So fish, pretty much as a rule, have oral jaws in the front of their face that are primarily used to capture prey. But they also have another set of jaws in the back of their throat called pharyngeal jaws. So these are modified gill arches that are functionally used to process prey or chew up prey or crush prey, as we'll see. And so even though fish have two sets of jaws, what's really unique about this species is it has two very different pharyngeal jaw morphologies. So these things here over to the right that kind of look like a slice of pizza, they're not a slice of pizza. They're actually the dorsal or prey's eye view of the pharyngeal jaw, which you can see laterally here in this anatomical diagram. But so in this species, you can go out to populations and you can find individuals that have these small little pointed teeth on their jaws that are called papilliform and are used probably to shred up plants or insects. But they also have another set of dentition called molariform teeth. So they're molar teeth, kind of like we have molar-like teeth. And so if you look at a population of this species, what you find is a highly polymorphic bimodal distribution in the size of the teeth. And that's what you can see here in this diagram. And so this species lives in this really unique habitat called Quatrocynicus. And in this habitat, there's well over 80 species that are actually endemic to this habitat. So there's somewhere around 12 species of fish that are endemic. There's endemic cacti, there's endemic freshwater shrimp here. There's several endemic mollusks that these molariform teeth are actually used to crush. And there's even an endemic box turtle that's aquatic. So there's an endemic turtle in this habitat. And what's really great about this system as well is if you look here in the center, this map to the left, out into the center is a mountain that juts out into the valley. And then around this mountain is a crescent of about 200 spring-fed pools. So springs that create these pool-like habitats called cienagas. And so this is a blow up of where the habitat actually is. So this is a picture of the USA, or Texas, actually where I'm from. This is the Rio Grande border. And then down about 400 kilometers south of Texas is this valley, Quatrocynicus. And this is just a few pictures down here on the bottom of some of these beautiful pools that look like crystal clear Mediterranean water, and where you can find all these assemblages of species. And so one of the things that's really nice about this habitat is it kind of gives us this replicated framework to look at the evolutionary ecology of some of these organisms. And so when I say that this polymorphic species is the same species, I think it's important to say why we think it's the same species. And so when I started working on this system, what we knew about the genetics and differentiation of these different morphs is that we knew that they actually interbreed in the wild. So people have gone out to these pools, they find them, they interbreed in the wild. We also knew, based on a bunch of allozyme loci from early genetic studies, that they were in Hardy-Weinberg equilibrium. And even though there doesn't seem to be any clear differentiation in their genetics, it's clear there is both a plastic and a genetic component to their pharyngeal phenotype. So if you raise these big enlarged teethed individuals in the lab, they actually are quite plastic. So they don't develop these big enlarged dentitions. And that's likely because of the snails, which I'll talk about briefly. So we know a lot about the ecology of Menglia and this system in Quatrocynicus. I've been involved in several of these studies, so we know that they are different in their diet. So the malariforms here are shown in black, and that's snails, so they eat snails. The other morph doesn't really eat snails. It seems to be specialized more on plants. There's also reduced competition when you raise these morphs together instead of with their same morph. We also know a lot about the community ecology of these fishes. So they clearly interact with the snails, and there's this negative correlation where you see that basically the papilliform, the pointed teeth individuals, are much more common, where the snails are much harder. So that's what's supposed to be shown on the y-axis with that diamond snail or whatever. And so in the populations where these snails are actually have much more robust snail shells, you get more of the papilliform morph. You don't get more of the malariform morph. Also, one of my favorite studies, and I'm not involved in this, but I think it's an unappreciated study. So one of the cool things about these habitats too, for any of the geologists in the room or paleontologists, is there are actually stromatolites in these freshwater systems. And so these were the major kind of encrusting coral-like structures before the evolution of metazoans in the world. But they now only occur in a few very select places, and Quatrocynicus is one of those places. And one of the reasons we think stromatolites disappeared is because when metazoans evolved to graze the stromatolites, it basically destroyed them as encrusting organisms. But one of the ideas is that this snail crushing fish basically interacts with these snails, and so keeps the snails at low enough abundance that these stromatolites can actually grow. So there's this indirect trophic cascade that's involved with the fish, the snails, and the stromatolites. But one of the things I started working on when I worked on the system is to try and tease apart some of the genetic basis of this differentiation. And so Minglia, again, is completely endemic to this valley. And so what I did was most systematists would do at the time is actually sequence some mitochondrial DNA from the two different morphs. And what I'll show you is this haplotype network that you see here at the bottom that's taken from all over these habitats that are color-coded in the map there. And the gray haplotypes are the malariform individuals, and the white haplotypes are the papilliform individuals. And this is more or less what you see when you do this kind of work. So this haplotype network here, what you can see is that the biggest haplotype that's most common is about 50-50 the papilliforms and the malariform individuals. And this is true for any one of these haplotypes. But what was really bizarre when we did this is there was a really divergent haplotype that was present in both the malariforms and the papilliforms at about 30% frequency. It's actually 4% sequence divergent between all the other haplotypes. And that's what's shown here with this little arrow. And so there's this really divergent haplotype, and we didn't know what was going on. It just seemed completely odd. But then I also worked a lot in Mexico in general and some of the other groups of fishes. And so that's what's shown here in this map. And we collected those fishes, and what we found was that this really divergent haplotype was actually identical to another species of Herikthes, the Texas cichlid, which doesn't normally or naturally occur in Quatrocynicus, but occurs all throughout Texas and well into southern Mexico, and abuts the distribution of Menglia here in Quatrocynicus. And what we know now is that Cyanogatetus, this other species, the Texas cichlid, has actually invaded Quatrocynicus through probably freshwater canals, because this water out in the middle of the desert is really valuable, and people have done a lot of water extraction, of course, and that's allowed invasive species to actually invade this habitat, and of course, destroy some of the freshwater pools as well. And so what we wanted to do was further investigate the genetic basis of this. And so this is a study based on rad tag analysis, where we took a bunch of Cyanogatetus Texas cichlids from outside the valley, and then a bunch of Menglia from 2 different habitats. And that's what's shown here across the x-axis, the malariforms and the papilliforms, and then also categorize these malariforms as either having the mitochondrial haplotype from the Texas cichlid, or the mitochondrial haplotype that is the Menglia mitochondrial haplotype. And so we did that with both morphs, and what we expected is some evidence of hybridization. And it's possible that this polymorphism might just be massive introgression from this Texas cichlid. That's what we thought. But when we did this, what we found is there was almost no evidence of introgression in any of either one of the morphs. So basically the nuclear genome, and we had about 6,000 SNPs at this point, just spread throughout the whole genome. And that's what's shown with this big block of red. Basically no real evidence of introgression here. But I wasn't satisfied with that. And so what I wanted to do was actually identify what the genetic basis of this polymorphism is. And so what we did is we basically hybridized a Texas cichlid with enlarged teeth and a Menglia with these papilliform teeth. And we generated a F2 generation QTL cross, and then genotyped those with rad tag markers as well, and then reconstructed the linkage groups and ran a QTL analysis to see what the genetic basis of this trait was. And what we found was one QTL that was really highly associated with the size of these teeth in these fishes. And so what that suggests is there may be a single gene that's actually involved in this polymorphism. And so one of the things I did while I was in Germany is with the Max Planck Institute in Dresden, we actually produced a really high quality genome of the Texas cichlid, Herikthes cyanogatetus. And then we resequenced the genomes of about 50 malariform and 50 papilliform Menglia, and then ran a GWAS analysis to see if we could pinpoint the region that's actually involved in this polymorphism. And so what we might expect from this kind of bimodal polymorphism is there is, again, a single large peak of association in the genome that's associated with these 2 phenotypes. And what's super cool and comforting in a way is that this peak of association is directly under the QTL peak. And what that suggests to me is that hybridization might be what's driving this single locus differentiation between Menglia and Cyanogatetus in terms of what's making this polymorphism present in Quatrocynicus. And so now that we understand the genetic basis of this, what we know is that water extraction and invasive species are really threatening this entire ecosystem. And it's clearly having an impact on a lot of the endemic species like Menglia. And so what I just want to put out there to everyone, how does this genomic understanding of this trait divergence that we see in this polymorphic species, that's really an iconic species of this habitat, really alter our understanding of things like population divergence? I mean, what we're seeing, I think, is the collapse of 2 species, not speciation, which is what I originally thought I was studying. Also, the community ecology, the relationships between the abundance of these morphs and the snails, it really flips on our head, I think, how we think about what's driving what in that interaction. And then, things like these indirect interactions of these trophic cascades, I mean, it doesn't, in my mind, suggest, because the mollusk crusher is probably really new on the scene, that's probably not what's driving the maintenance of these stromatolites and these things like that. And then I think, if we want to conserve, the original species of Menglia, how do we actually do that if there's just a single gene that seems to be rapidly introgressing in these populations, and what do we actually do about the conservation of the system if we really care about conserving the native diversity? And so I think it offers an opportunity to really address a lot of potential complexities, and also an interesting way that, understanding the genomic basis of this trait really might inform our conservation priorities and these type of things. So I just want to leave you all with that, and take any questions, or provide any comments. So thank you.
Thanks very much indeed, Darrin. That's fascinating stuff. Does anybody have any questions that they'd like to ask or comments? So I do have some questions, but they're more to do with the ecological side than the genetic and evolutionary side. I was interested in the water extraction and the invasive species side of things, as well as the kind of community dynamics, I guess. But could you elaborate a little bit more on that kind of water extraction? Is it accelerating? Is there an increasing pressure to take more water out of the system? And is there a strong pushback by conservation agencies, or is it looking like it's just going to roll away from?
No, I mean, so it's, all those things are part of the equation here. So what we kind of know, and this is based on very spotty records, is that the water extraction probably began back in the 1800s with some canals that are still around, and we know that they're still around. And so it's been extracted for a long time. The thing about this system is that the salinity for a freshwater system is really high. And it's probably one of the things, these snails, if you look at them, they look a lot more like marine snails than freshwater snails. And that's probably because of the calcium carbonate availability and these kind of things, because of it's so high in salinity and dissolved salts. And there's absolutely gypsum sand dunes. You can see gypsum there. So there's some all kinds of calcium availability in the water and things like that. So the extraction's been going on a long time, but it's not that efficient, because it basically you're pouring salt on your crops. Right? That's not good. And so, but what has happened, because people recognize this for a long time, is that having a bunch of unique species. So there's been protection in the valley for quite a while. It hasn't really stopped the extraction of water directly at surface waters, but it has sort of slowed that down. But the problem is the water table. So in the surrounding areas of the valley, for instance, the biggest milk producer in Mexico, called Lala, Lala has actually been drilling wells of water that's not so salty, that's right outside the basin, and pumping that water out, producing alfalfa crops and fields, feeding that to cows for their milk in the middle of the desert, right? And this is causing the water table visibly over like the decade, for sure, to drop. And there are places in the valley where you can see like these dried stromatolites look like, giant outcrops of there were used to be pools or spring-fed pools. So all of that's happening, and there is a wide recognition of the valley as being a unique place, and a World Heritage Site even. And so there is a lot of will and and effort to try and protect the area. Of course, another thing that's really complicated is the last time I was there was, I think, 2011. And since then, like, the narco trafficking has just exploded. So in Mexico in general, but this area in particular. So efforts to work there are difficult. And so that just adds this whole other layer of complexity on trying to promote some kind of protection, do research on the valley, these kind of things. So, it's a geopolitical, like, crazy place, So, and so, so all of these things are completely involved. I mean, I don't think you can think of one completely without the other. I mean, like, it's a lot of complex problems. I mean, there's a lot of factors here at play, and ruling on groundwater in a place like the Chihuahuan Desert in Mexico is difficult. I mean, and politically charged too. So
imagine it is, yeah.
Yeah.
Oh, that's interesting. The narco trafficking kind of perspective, that does add a whole other layer into that. But it's not enough to kind of prevent the activities that are causing the problems.
No, not really. I mean, you know, things,
yeah.
there's bribes, people, you know, people are protected. And, you know, this is on one of the main highways where a lot of the narco trafficking actually occurs.
Mm-hmm.
And so it's probably pretty bad. And I mean, I haven't been there a while. It's been so bad, but, you know, but it's bad.
Yeah.
So,
yeah. Because I mean, sometimes things like that actually help to protect the natural environment, because people don't go there and cause it problems. But it doesn't sound like that's going to be the kind of small silver lining that there might be in this situation.
Right. Right. Well, maybe.
Does anyone else have any questions to ask? The other
Can I ask
area,
a quick question? Sorry.
please, Adam. Yeah, go ahead. Yeah.
Is there any cost to developmentally for the fish to have the different dental morphology? Just like having, I thought maybe the molar one seems to have just more bone to it, and that might be an extra cost exacted on it.
Right. I mean, it could. I mean, in this place where there's so much calcium again in the water, and that may be one of the things that kind of ameliorates the cost of building these big bones and big teeth and replacing their teeth. And fish replace their teeth throughout their life. I mean, that's one of the things I didn't mention, but you know, like sharks that replace their teeth. I mean, we're the weirdos that only replace our teeth one time, right? So fish are the normal ones. But it probably is somewhat costly. Eating these snails, also, they break their teeth fairly frequently. But because they replace them all the time, it's not as costly as it might be for us. The genetics of this are interesting, because what it seems like is we know more or less what the gene is. So it's in this small little gene cluster called Iroquois. And we know those genes are actually involved in a host of different phenotypes. Your heart, actually, these genes are expressed in your heart. They're expressed in muscles and your lungs even. And so what seems to be the case is there's actually maybe a homozygous lethal situation going on that may maintain the polymorphism itself, so that if you get both alleles of the of the Menglia, I'm sorry, then you actually might die. And so the malariform guys, the guys with the big teeth, are actually heterozygous always at this locus. And the other morph, the Menglia morph, is always homozygous for the Menglia alleles or whatever. And so that, like, lethal homozygosity is another thing that's maintained in the polymorphism probably pretty clearly. And so we're just now getting a handle on that. But that's by looking at the GWAS and knowing the annotation of the genome, which we've just recently completed. Like, that is something that seems to be going on. So it is costly, probably in a serious way, to be homozygous, but the exact dynamics of that are something we're still kind of trying to get a handle on.
Cool, thanks. And just to kind of follow up there, it just dawned on me, is there any possibility that they can change the dentation as they replace, like in line with the snails that are available in the system?
Right. That is actually something that people have asked a lot. I do know two things that suggest they can't. So one is I've looked at individuals that are very, very small, like before they ever start crushing snails. So even as small as like 20 millimeters, they still have big malariform teeth or small teeth. Also, I've done a lot of CT scans, like looking at the replacement of the teeth. And it doesn't seem that in any of the individuals, and I've looked at probably like 30 of each morph, basically none of the individuals are replacing, say, the big teeth with really small teeth, or the small teeth with the really big teeth. So it seems to actually be set up very early in development and ontogeny. And it doesn't seem to flip back like that. But that's one of the things that I definitely thought was possible, maybe. it doesn't seem to be that way.
Cool. Cheers.
Yeah.
Great talk.
Thank you.
I was going to ask something similar about the kind of the balance between the two morphotypes and whether that shifts through time in a way that's potentially linked to the availability of prey, which could I guess could happen quite quickly if they are able to switch during their own lifetimes, but would take longer, I guess, if it was a population level kind of effect, right? Is there any evidence of that kind of long-term changing in the balance between the types?
Well, there's a huge amount of variability in the frequency. So there are some pools, and that's what's great about having the different pools and and actually the metapopulation aspects of everything. I mean, it's why I originally got into this, not because I'm like a tooth but there's a huge amount of variability in that, and it does seem to be related in part to the hardness of the snails, but that's also kind of related to the productivity in the environment. So in some of these environments, there seems to be really high productivity. There's a lot of plants for the snails to eat, and a lot of available nitrogen and things like that. So the and I don't really know, stable isotope and geochemistry that well, but there is a lot of variation in that, and that seems to be driving the hardness of the snails, which seems to be interacting with the frequency of the morphs. So there is some like sort of ecosystem level processes that might actually be even driving some of this or feeding back into it at least. So there is some of that. We haven't, it's one of the things that I wanted to spend a lot of time actually uncovering, but again, that's when they started chopping heads off. And
the circumstances made it difficult to do that. It's a real shame, isn't it? Because it's a fascinating system altogether. Does anybody else have any questions? We're just about to run out of time, so I'll give others one last opportunity to come in with a question. And if there's nothing, then I'm sure, Darrin, you'd be happy to receive any questions any other time and be open for collaborative opportunities. And I guess that's kind of one snapshot of your range of interest, but I guess you're doing a lot of other things along similar lines in other contexts, right? And
exactly
open to collaboration.
For sure. Yeah, I'd be happy to take any questions anybody has. Please ask away. I love talking about fish teeth and.
As you do.
Okay.
Brilliant. Thanks a lot, Darrin. And
Thank you
thanks
all.
everyone for coming along. That was great.
See you.